In this lesson, we're going to be looking at how bacteria control how much protein they make from their genes. When we talk about regulating genes, we're thinking about how the cell controls how much protein is made at a certain time. Because manufacturing proteins is a multi-step process, there are several different ways in which bacteria can control how much of that protein is ultimately produced. Imagine for a minute that you're a farmer. There are lots of things that you could potentially grow: wheat, corn, rice, vegetables. If you weren't thinking about exactly what you would grow, you might be tempted to get all the machinery, seeds, and other utilities in order to grow any of those vegetables. However, if you then decided just to grow rice, then your combine harvesters and other machinery that isn't useful for growing rice would go to waste. Clearly, it would be a better move to only buy the materials that you need for the crops you plan to grow. It's simply wasteful to buy all that extra stuff. Similarly, bacteria make similar decisions. For example, if they're growing on the sugar lactose, then they will need enzymes in order to process the lactose. However, if there's no lactose in their environment, purchasing or making those enzymes is energetically unnecessary and wasteful. Therefore, bacteria will tend to only make the enzymes they need and are suitable for their environment, even though they're capable of genetically producing others. Shutting down production or ramping up production of particular proteins can be done at any stage of the process. In transcriptional control of gene expression, the cell controls how much and when mRNA is produced. This is a very energetically cautious way of controlling gene expression because production of mRNA and proteins requires a fair amount of energy. If the cell stops that production right at the first step, then its energy costs are minimal. This is like you, the farmer, deciding to only buy the equipment that you actually need. However, if a cell suddenly finds itself in an environment where it needs that particular protein, then it will be a while before the protein can be made, as the cell needs to generate enough mRNA through transcription, generate protein through translation, and then that protein may need to be activated. If you, the farmer, suddenly decided that you wanted to grow wheat, then you'll need to go shopping for a combine harvester, and that can take some time. The cell can also control the translation of mRNA into protein. This requires sort of intermediate energy use and an intermediate time in order to activate proteins that are made. Finally, cells can manufacture proteins that have them sitting around waiting to be used. This is energetically more wasteful because the proteins are produced whether or not they're needed and may eventually be degraded without ever being used. However, having the proteins lying around may be useful because the cell is able to activate them and use them on very short notice. Again, this is similar to you having bought all of the possible farming equipment, and if you decide to plant wheat, well, you'll have a combine harvester. If you plan to plant rice, then you have all the materials to do so, and you don't need to spend extra time shopping. In previous lessons, you learned about feedback inhibition, and this type of regulation is really common in metabolic pathways in bacteria. Remember that with feedback inhibition, the end product of a biological pathway is used as the regulatory molecule and feeds back into a previous step to regulate that pathway. In this case, tryptophan is the final molecule produced, and tryptophan can feed back to inhibit an enzyme along the pathway. When tryptophan acts like an inhibitor like this, a buildup of tryptophan will reduce the amount of tryptophan that's subsequently produced. As the levels of tryptophan decline, then there will be less inhibition and ultimately more tryptophan will be produced. Similarly, tryptophan can act as an inhibitor at the genetic level to reduce the amounts of transcription and translation that occurs when there's lots of tryptophan present. Again, as tryptophan levels decrease, the inhibitory action of tryptophan will be reduced, and more transcription and translation will result in more enzymes producing more tryptophan. In bacteria, a common feature, particularly of metabolic genes, is that they're all located adjacent to one another and all have the same point of control. In the tryptophan operon shown here, you can see that there are 5 genes adjacent to one another. These genes are separated by stop and start codons that allow for the translation of protein. In addition to the genes, there's a single promoter that tells the RNA polymerase where to bind. Once bound to the promoter, the polymerase will make a single mRNA transcript, and this transcript will then be separately translated into the 5 proteins. Part of the promoter is what we call an operator. This is the sequence of DNA that influences or controls whether or not RNA polymerase is able to advance and transcribe the gene. In the case of the tryptophan operon, the operator is able to be bound to a repressor protein. This repressor protein is able to recognize the DNA sequence of the operator and bind to it. However, it only binds in the presence of tryptophan, which is called a co-repressor. When the repressor is made, it's in an inactive form and can't bind to the operator. However, in the presence of tryptophan, its shape changes and it's capable of binding to the tryptophan operator. When it's bound, the RNA polymerase is unable to advance from the promoter sequence to the genes, and thus the repressor represses transcription. Let's take a moment to think about how this system works under different conditions. If the levels of tryptophan are high, then tryptophan will most of the time be bound to the red repressor. When bound to the repressor is active and can bind to DNA. When it's bound to the operator, the RNA polymerase can't advance, so no mRNA is made, and therefore no enzymes are made. Finally, because no enzymes are produced, tryptophan production declines, and eventually the levels of tryptophan will probably start to decline. When the cell is running out of tryptophan and needs to manufacture more, there will be very little or none bound to the repressor. Without tryptophan bound to the repressor is inactive and it can't bind to the operator. With no repressor bound, the RNA polymerase is able to advance, make mRNA, and ultimately the levels of tryptophan will then increase. Before we leave this, let's look at the repressor itself. The tryptophan repressor is a protein and is therefore manufactured as a result of transcription and translation. The tryptophan repressor gene is always turned on, and there's therefore always repressor molecules present in the cell. The tryptophan repressor gene is not part of the tryptophan operon and may be located at a totally different region of the chromosome, or even on a separate chromosome if the bacteria has more than one. In many ways, the lac operon, which controls the metabolism of the sugar lactose, works in similar ways to the tryptophan operon. As an operon, all of the necessary genes are located together on the chromosome, and there's a single point of regulatory control. In this case, however, the repressor is active in the absence of a co-repressor. Notice again that, like with the tryptophan operon, the regulatory gene is always turned on to produce the repressor, and that regulatory gene is outside of the operon. In the absence of lactose, the repressor is active and binds to the operator. With the repressor bound, the RNA polymerase is unable to advance, and so no RNA for the lactose gene is produced. With no mRNA made, there will be no enzymes, and the cell will not be able to metabolize lactose. However, notice that this is exactly what the cell needs. It would be wasteful of the cell to produce those proteins in the absence of lactose. And so by repressing their production, the cell becomes more efficient. When the cell's in the presence of lactose, an isomer of lactose called allolactose acts as an inducer, and it binds to the repressor protein. When bound in this way, the repressor is now inactive and can no longer bind to the operator. With no repressor in the way, the RNA polymerase is able to advance and transcribe these genes necessary for processing the lactose. These genes allow more lactose to enter the cell, and once inside, enable the cell to use the lactose for energy. The operon contains 3 important genes. LAC-Z produces a protein called beta-galactosidase, which breaks lactose into its 2 component monomers. LAC-Y produces permease, which allows the cell to take in lactose. The third gene, LAC-A, is used to transport sugars out of the cell in case they become so concentrated that they're toxic to the cell. Finally, LAC-I is the regulatory gene that produces the repressor. It's located outside the operon. You can see that when the cell is in the presence of lactose, it produces the enzymes needed to process that lactose. But in the absence of lactose, it does not waste its energy producing enzymes that are of no use to it in that environment. Now, some of the words we use to describe regulation can be pretty tricky. We say that the LAC operon is inducible because by itself the repressor is active, and it needs something to happen to it, something to bind to it in order for it to break free of the operator and allow transcription to happen. Therefore, the default is for the gene to be turned off, but the gene can be induced to become active. The tryptophan operon, however, has a repressor that's inactive by itself, and the gene is therefore usually turned on. Because the addition of tryptophan represses the transcription of RNA, we say that it is a repressible operon. Inducible operons are relatively common in catabolic pathways, which are used to break down nutrients into their component parts. A cell only needs these proteins sometimes and can therefore be induced to produce them. Repressible operons are common in anabolic pathways, which are pathways that build up compounds. The default of the cell is to make these proteins, but if not needed, their production can be repressed. Now, both of these examples are examples of negative regulation. We call this negative because both operons are switched off by the active form of the regulatory molecule. Let's look at that again. In the LAC operon, the operon is active unless the repressor is active. Therefore, the operon gets switched off by the active repressor. In the tryptophan operon, the operon is also active unless the repressor is active. In other words, the operon gets switched off by the active repressor. The difference is what makes the repressor active. In the LAC operon, it's active by itself, but in the tryptophan operon, it's active only when bound to the tryptophan. So again, these are both examples of negative regulation because the operon gets switched off by an active repressor. One thing you should think about, since we've already learned about how genes operate, is what would happen if certain mutations were to happen in the gene. We'll walk through mutations in these LAC genes, but I encourage you to think about similar mutations in the other operons. So for example, what would be the effect of a frameshift mutation in the LAC-I gene? To think about this, remember that a frameshift mutation is likely to render the protein useless. If the LAC-I gene doesn't work, then it will never bind to the operator. If it never binds to the operator, then the polymerase is free to transcribe the downstream genes, even without lactose present. If there's a mutation in the LAC-Z gene, the lactose could enter the cell, but wouldn't be cleaved into monosaccharides, and therefore couldn't be used as an energy source. Finally, a mutation in the LAC-Y gene would make it so that lactose could never enter the cell because permease wouldn't be active. Again, in negative regulation, the regulatory protein becomes active and stops transcription. Here, you can see that when there's no negative control, transcription occurs, but with negative control, the repressor represses transcription. Positive control, on the other hand, is the case where the regulatory protein triggers transcription. In other words, there's little to no transcription without the regulatory protein, and once present, transcription is able to happen. Now, we've used the LAC operon as an example of negative regulation. In fact, it's a bit more complicated than that because the LAC operon is also under positive control. Let's see how that works. When an E. coli cell is in the presence of both glucose and lactose, the LAC operon remains off, and the cell metabolizes glucose, its preferred carbon source. It's only when both glucose levels are low and lactose is present that the LAC operon is turned on. So remember, glucose is an easier molecule to break down and obtain energy from, so the bacteria prefer glucose. When an E. coli cell is in the presence of both glucose and lactose, the LAC operon remains off, and the cell metabolizes glucose, its preferred carbon source. It's only when both glucose levels are low and lactose is present that the LAC operon is turned on. So remember, glucose is an easier molecule to break down and obtain energy from, so the bacteria prefer glucose. It's only when glucose is not available that the cells turn to other sources of carbon, such as lactose. If glucose is available, then they will always use it. When glucose levels outside the cell are low, there's an increase in the concentration of a molecule called cyclic AMP. Some of you will recognize cyclic AMP as being a secondary messenger involved with many signaling pathways. At any rate, as the glucose levels drop, cyclic AMP increases. Cyclic AMP then binds to a protein called CRP, or often called CAP. When it builds up in the cell, cyclic AMP will bind to CAP, and CAP will bind to the promoter of the operon. When it's bound to the operon, it increases the affinity of the polymerase for the LAC promoter. Effectively, CAP, bound to cyclic AMP, increases the transcription rate. So notice that we're using this as an example of positive regulation. The regulatory molecule, in this case cyclic AMP, turns on transcription of the gene. The regulatory molecule has a positive effect on transcription. Notice, however, that in order to transcribe the gene, the LAC repressor must be inactive. If the repressor is blocking the polymerase, transcription will not proceed. Therefore, lactose must be present, but glucose must be scarce. On the other hand, when glucose levels are high, cyclic AMP remains low, and as a result, there's no activation of the polymerase. If the polymerase hasn't bound to the promoter, it won't produce any of the mRNA. Without these genes turned on, the cell primarily metabolizes glucose, even when it has access to lactose. One way you can think about these two different bits of regulation of the LAC operon is that the LAC repressor is like an on-off switch. If the repressor is active, there can be no transcription until the repressor unbinds from the DNA. The cyclic AMP cap protein complex sort of acts like a dimmer switch, increasing transcription when cyclic AMP levels are high and reducing it when cyclic AMP levels are low. Another example of an operon with positive control is the Arabinose operon. As with other operons, the genes that are transcribed are all used in the same metabolic pathway. Thus, ARA B, A, and D are genes that get transcribed and then translated to make proteins for the pathway. The regular protein here is ARA C, and that is coded for by a gene nearby, but not part of the operon. When Arabinose is present, it binds to the ARA C protein, and that has a positive effect on transcription. Strangely, ARA C can act as a repressor as well as an activator. When there's no Arabinose present, the ARA C protein actively represses transcription by having two parts of the ARA C dimer bind together. By binding together like this, they form a loop in the DNA and effectively prevent polymerase from having access to the promoter. This further reduces the amount of transcription that happens in the absence of Arabinose.